Swayam Prabha. Digital India. Educated India.
So once again, good day and welcome to today's session on skin and fascia. This is Dr. Mauna Subbaramaya, working as Associate Professor at Kempegowda Institute of Medical Sciences, Bengaluru. Skin, as we all know, is a humongous topic. So I will not be doing justice in saying that I will be covering all aspects of skin in today's lecture. So this particular session is primarily aimed at overlying or broadly outlining the general aspects of skin and fascia pertaining to its functions, a few layers, the cells, and the modifications and general features of fascia. So let's begin the session with a riveting activity. So I'm going to portray a few pictures. So you will have to tell me what is common between these pictures. In other words, find out the analogy between the following pictures. So here goes your first picture. The next picture is this. And the third one is this. So could you find out any common terminology which could connect all the three pictures? No? So here's a clue for you. The first picture suggests infection, that is herpes infection or herpes zoster, otherwise commonly referred to as shingles. So yes, so we are talking about dermatomes. So what are dermatomes? Dermatomes are segments of skin which are supplied by a typical spinal nerve. So when I'm talking about dermatomes, each of these pictures have a connection to this particular word. Of course, the third picture is just a portrayal of the dermatomes themselves, so it is self-speaking. Number 30 correlates to the number of dermatomes in our body. That is one less than the typical spinal nerve number, because C1 does not have any cutaneous distribution. And the reason I put up shingles is the distribution pattern of this particular infection is based on dermatomes. It affects about 2 to 3 adjacent dermatomes. So with this activity, I think you are stimulated enough to proceed to the core of the lecture. So here we are. We will be talking about the skin, or briefly introducing the skin pertaining to its functions, components, development, differences between thick and thin skin, the nerve supply, blood supply, lymphatic drainage, and a few clinical aspects. So skin as such is the biggest organ or the largest organ of the body. It is the largest sensory organ too. It contributes to about 16% of the total body weight and 8% of the total body mass. And if we have to talk about the surface area of skin, or accounts for about 1 to 2 meters square. And the thickness of the skin varies from 0.3 mm to 3 millimeters. Okay. So this is a brief numerical facts in relation to skin. So coming to the functions of skin. So why do we need this protective covering? So like I said, the first function is protection. So it protects us from physical, chemical, and biological hazards. So it protects us from the UV rays, from acids, chemicals, and invasion of microorganisms. So it forms a nice barrier between the internal and the external environment of the body. The next important function is thermoregulation. This of course is more so in the lower animals, where they are coated with a thick fur of hair. In case of humans, though the hair is minimal, There are other aspects which bring about thermoregulation, like the amount of blood flow through the capillaries, the activity of the erector pili muscle, which brings about the erection of the present hair, resulting in conditions like goose flesh, and also through evaporation of sweat, which causes cooling of the body. So, in these ways it brings about or rather plays a role in thermoregulation. The next important function of course is sensation. So, in fact skin is the largest sensory organ, and more so in humans because it is hairless compared to the lower animals, and it is predominantly a sensory in function more than the thermoregulator. The metabolic activities of the skin includes production of certain hormones, vitamin D, and also the certain secretions which are poured out by the skin, that is through the sebaceous and the sweat glands. They act as pheromones. So, all these factors which are liberated by the skin bring about certain metabolic activity in the skin. There are some excretory function. Unwanted waste materials are thrown out in the form of sweat. And last but not the least, identification. So, we can identify a person based on its color, hair texture. So, there are various modalities, and fingerprints is the main identifying modality which is used even by forensic experts. So, the dermatoglyphics, which involves the study of fingerprint pattern, it is unique to the individual. So, no two identical twins have the same fingerprint pattern. So, in this way skin acts as a vital organ in identification of a person. So, these are some of the general functions which the skin performs. So, with this we will move on to the structure of the skin. So, these are the basic components which make up the skin. The first layer is the epidermis. So, this is the outermost layer. It is a predominantly cellular layer with very little intercellular substance. The next layer is the dermis. So, this is a predominantly connective tissue layer which is made up of a number of connective tissue fibers, that is the collagen, elastic and reticular fibers, and also has all the other components of regular connective tissue has. So, beneath these two layers, which are the primary layers of skin, we have the hypodermis, or commonly referred to as the subcutaneous region, which can also be synonymously used as the superficial fascia. So, all these three words, that is hypodermis, subcutaneous region or superficial fascia, almost all mean the same. So, they are present beneath the skin. Now, associated with these two layers of skin, that is the epidermis and dermis, there are a few other structures which are referred to as appendages of the skin. So, what are these appendages? They include the pilosebaceous unit. So, like the name suggests, it is made up of pilo means hair, sebaceous gland, and there is one more structure in relation to this unit, that is this particular muscle which is present, that is the erector pili muscle. So, all these three put together form the pilosebaceous unit. The next appendages we are talking about is the one which is shown in green color, and that is the sweat gland or the sudoriferous glands. And the third appendage is the nails. So, this is all modifications of these two layers. So, they are derived from the layers of skin, hence they are referred to as appendages of skin or the accessory units of skin. So, let us talk about these in a little more detail. So, coming to the epidermis, what are the layers of the epidermis? It has 5 basic layers. They are the stratum basale, stratum spinosum, stratum granulosum, stratum lucidum and stratum corneum. So, these are the 5 basic layers which are present in the epidermal zone of skin. Now, I will not be going into minute finer details of these layers. I will just give you a brief overview because they will be dealt in detail in histology sessions. So, what do we see in the stratum basale? So, this is a layer of columnar or high cuboidal cells which are predominantly mitotic. So, they give rise to the other cell layers. So, once the cell starts in the basale, it moves through all these layers and finally it is shed off in the stratum corneum zone. So, this transition of the cell from the basal layer to the superficial zone is referred to as a epidermal turnover rate, and this anywhere varies between 14 days like as seen in the forearm, or on an average it takes about 45 days. In case if this epidermal turnover rate is higher than usual, then it can result in excessive scaling, which can lead to a condition called as psoriasis. Okay, so, that is the mitotic activity which we see in the stratum basale. The next layer is the stratum spinosum. So, as the name suggests, the cells here look very spiny in nature, or they have spiny processes or dendritic like processes. So, why do we get this is during preparation of the routine histological slides, the cell contracts and the edges of the cells, they are in contact with the adjacent cells through tight junctions. So, these tight junctions are preserved and they give a spiny appearance to this zone. This is also called as a Malpighian zone, and some authors they consider the first 3 layers as the Malpighian zone itself. The next layer is the stratum granulosum. So, as we come from the stratum basale to the stratum spinosum, the cell size, the height reduces. So, it is initially columnar, then it becomes polyhedral, then it goes on flattening. So, as we reach the stratum granulosum, it is a little more flatter compared to the spinosal layer. And here, as the name suggests, the cells have typical granules in them. And what are these granules made up of? They are keratin granules. So, when we are talking about these granules, we have to talk about a little bit about the keratinization process as well. So, this keratin is nothing but a protein filament which is started in the region of the stratum basale. So, cells here they start producing the keratin which get aggregated to form filaments in this particular zone, that is the spinosum zone, and then they are further aggregated to form granules in the stratum granulosum. And as we proceed more superficially through the stratum lucidum, they become more and more, and they are finally shed off along with the dead cells of the stratum corneum, and they form a nice waterproof layer over the skin. So, this is called as keratinization, where the protein keratin is being formed from the stratum basale and finally reaches the stratum corneum. And if there is excessive production of keratin associated with excessive epidermal turnover rate, we have the condition that is mentioned earlier, psoriasis. So, this is about briefly regarding the layers of the skin: stratum basale, stratum spinosum, stratum granulosum, lucidum, and finally the stratum corneum, where the cells almost are flat and they are dead without any nuclei, and they are present along with the keratin filaments. And in the lucidum area, the layers are, the cell membranes are very indistinct. There is a nuclei, but they are very pycnotic in appearance. So, this is a living layer, but the cell membranes are very indistinct, and this layer seems to be absent in thin skin, or even if it is present, it is present as a very thin zone. Okay. So, this is about the layers. Coming to the cells which are present in skin, the majority of the population forms the keratinocytes. So, these are present throughout all these 5 layers. They start in the stratum basale, go on to the spinosum, accumulate keratin in more amounts and reach the granulosum. Then their cell membranes become indistinct, and they finally shed off along with the keratin and form the stratum corneum. So, these keratinocytes, they are small polygonal cells with few dendritic processes, and they start accumulating keratin, producing keratin and accumulating them as and when they transit through the different layers of skin, and they form the majority of the population of skin. The next cell is the melanocytes. Of course, this is responsible for the color of the skin, and the number of melanocytes is quite constant in different sexes and races. However, the pigments which are contained in intracellular organelles called as melanosomes, those vary. So, the amount of melanosomes will determine the color, and the amount of melanin which is present will depend on other external factors as well, like the UV light. Then also it depends on the amount, the nature of hormones, etc. So, various factors will determine the amount of melanosomes which are present in these cells, and melanin can be present in the form of eumelanin or pheomelanin, which is responsible for the reddish and the dark brown complexion. So, this is about the melanocytes, and again, like the keratinocytes, these are also predominantly present in, they are having dendritic processes, and they are predominantly present in the stratum basal zone. The next variety of cell is the Langerhans cell. These cells are defensive in nature. In fact, they form a part of skin associated lymphoid tissue, SALT that is called. Okay. So, they form an important aspect of the defense mechanism, and they are present throughout the layers of the epidermis, and their processes extend beyond one or two cells. So, they have long elongated dendritic processes, and these cells they have certain intracellular bodies called as the Langerhans bodies or Burbeck's bodies. Of course, the function is yet to be known, and these cells are primarily responsible for conditions like allergic contact dermatitis, or they are also responsible for graft rejection. And the last variety of cells which we come across in the epidermis is the Merkel cells. So, these are predominantly present in relation to the basal layer, and they extend their processes between the stratum spinosal cells as well, and they are sensory in nature. Okay. So, these are the 4 different types of cells which we come across in the epidermal zone of the skin. Next, we will move on to the dermis. So, the next underlying layer is the dermis, and this dermis has 2 layers, that is the papillary layer and the reticular layer. So, the papillary layer is the intimate layer which invaginates into the epidermal layer. So, there is an interdigitation of the epidermis and the dermis. Now, the reason for this peg-like attachment between the dermis and the epidermis is to prevent it from slipping off. If the separation was uniform, if one layer was placed over the other, then there would be chances of peeling off the dermis from the epidermis. So, this peg and socket like of arrangement will prevent it or increase the chance of attachment between the epidermis and the dermis. So, that is the papillary layer into which the nerve plexus and the other blood vessels will form loops and enter. The next layer is the reticular layer. So, that is made up of typical connective tissue zone having all the major nerve plexus, blood vessels and the connective tissue cells. And in fact, leather, what we speak about is nothing but a tanned layer of the dermis. So, what does the dermis comprise of? It has cells, that is the connective tissue cells, which include the lymphocytes, the eosinophils, WBCs, etc. The fibers, that is the collagen fibers, elastic fibers and reticular fibers. Now, the collagen fibers arrangement in the dermis is typical in fashion. So, and they are arranged in regular bundles, and in different parts of the body they run in different directions, and these are called as Langer's lines, and they form the basis of many surgical incisions, because if surgical incisions are put along these Langer's lines, there is minimal scarring. But if they are put perpendicular to these Langer's lines, then there is more chances of scarring and delayed wound healing. So, collagen fibers in case of dermis, they play an important role in this cosmetic department. The elastic fibers of course maintain the elasticity of the skin, and as age progresses the elastic fibers reduce in number, and thus we have wrinkling of skin and so on. Okay. So, the other components of the dermis include the capillaries and the nerve plexus. So, this was about the dermis, that is the two layers, that is the papillary layer and the reticular layer, and the different components which include the cells, fibers, capillaries and the nerve plexus. So, this was the second layer of skin. So, now moving on to the appendages of skin. So, like we spoke about, there are three primary appendages of the skin. The first one is the pilosebaceous unit. So, what is this pilosebaceous unit made up of? So, this is nothing but the sebaceous gland, this is the muscle and this is the hair follicle. So, all these three put together they form the pilosebaceous unit. So, sebaceous glands, they are usually associated with hair follicle. Of course, we have a few sebaceous glands which open directly onto the surface, for example, around the lips, then in genital areas, in prepuce, in clitoridis, etc. So, here they directly open onto the skin, but elsewhere they are always associated with a hair follicle, hence they form a part of the pilosebaceous unit. And each sebaceous gland is a cluster of cells present as alveoli, okay? And the superficial cells of these are flattened in nature, but as you go from the superficial zone to the inner aspect, the cells become more and more rounded, and the centralized cells, they are filled with secretions which are poured onto the hair follicle. So, the secretion what they discharge is referred to as the sebum, and it is very oily in nature, and it maintains the shininess and it forms a nice waterproof layer over the skin and hair. So, that was about the sebaceous gland, and this is a typical example for a holocrine gland. So, what is this holocrine gland? It means that whenever the cell has to release the secretions, the entire cell dissolves and it releases the secretion by destruction of the cell itself. So, that is referred to as holocrine. The whole of the cell gives out or pours out the secretion. So, that is about the sebaceous gland. Next, we have the erector pili muscle. So, this erector pili muscle is always in relation to the pili, that is the hair, and like the name suggests, it is erector. So, it makes the hair follicle stand erect. So, it is attached to the base of the hair follicle and the other end is attached to the lower part of the dermis. So, when it contracts, it makes the hair shaft stand erect. So, it plays a very important role in thermoregulation and heat control. When this happens, we have the goose flesh appearance of the skin. Then the final and the most important part of the pilosebaceous unit is the hair itself. So, the hair follicle, it has an outer component which projects outside from the skin, this is referred to as the shaft of the hair, and the portion which is embedded into the dermis. So, that is the root of the hair. So, in fact, all these appendages which we are talking about, like the sebaceous gland, the hair follicle, they are extensions of the epidermis itself. So, hair is nothing but an extension of the epidermis into the dermal zone. So, this part is referred to as the root and this part is referred to as the shaft. The terminal portion of the root is enlarged to form what is known as the hair bulb, and this is invaginated by the dermal papillae. So, this region is the invagination of the dermal papillae into the hair bulb. Now, if we take a section of the cross section at any level of the hair follicle, it is comprised of the follicle as such, surrounded by an inner root sheath, and which is in turn surrounded by an outer root sheath. Now, the hair follicle as such is made up of 3 layers. The innermost is the medullary zone, surrounded by what is known as the cortex, and this in turn is surrounded by a cuticular layer. So, the medullary zone is nothing but an aggregation of the keratin filaments with a few here and there living cells. The cortex has less number of cells, and in the cortical zone air bubbles start appearing as age progresses. So, it loses the color and shine of the hair because of these presence of these hair bubbles. And outer to the cortex we have the cuticular layer, which is nothing but cells, flattened cells which lie edge to edge over one another. So, this comprises the complete hair follicle. Now, surrounding this hair follicle we have an inner root sheath. So, the inner root sheath is in continuation with the cuticle of the hair follicle. Then this is surrounded by a layer called as Huxley's layer, which is about 3 to 4 layers in number, and this in turn is surrounded by a layer of cuboidal cells, that is the Henle's layer. So, these 3 units comprise the inner root sheath. Now, external to this we have an outer root sheath. So, this has again a few fibers and keratin filaments, and outer to this we have a glassy membrane which encloses the outer root sheath. So, in a cross section from internal to external we have the medulla, the cortex, the cuticle, the Huxley's layer, Henle's layer, outer root sheath and a glassy membrane. So, this is the structure of a typical hair follicle. So, that again is a pilosebaceous unit. The next appendage is the sweat gland, or otherwise called as sudoriferous glands. Now, each of these glands they have a coiled basal portion and an elongated conducting portion which pierces the epidermis and opens into the exterior. Now, there are 2 varieties of sweat glands. You have eccrine glands and you have apocrine glands. Eccrine glands, the cell does not alter in configuration while discharging their secretions, and they are present in relation to most of the areas, and it is predominantly seen in case of forehead, palms, soles, etc. And sweat glands are typically absent in case of wherever the sebaceous glands open directly to the skin. In those areas these sweat glands are minimal or absent, that is around the lips, around the prepuce, the genital region, that is the clitoridis, etc. So, we were talking about the 2 varieties of sweat glands, that is eccrine and apocrine. So, eccrine discharges without any disfiguration in the cell morphology, whereas in the apocrine part this is the atypical sweat gland where the terminal portion of the cell is discharged. So, apo, that is the apical part of the cell, goes off or while pouring out the secretions. So, there are two varieties of these present in association with skin, eccrine and apocrine. So, this is about the sudoriferous glands or the sweat glands. The last appendage is the nails. Now, nails are nothing but the cornified layer of the epidermis, which are protecting the terminal distal portions of the phalanx. So, they are a modified stratified corneal zone. So, when we talk about the nail, we have a lateral fold, skin fold, which covers the lateral aspect of the nail. Then we have the proximal skin fold, which projects or protects the proximal portion of the nail fold, that is the root of the nail. So, if we take a section of this nail, we find this portion of the skin which projects beyond the root of the nail. So, this region is referred to as the nail plate, and underlying this we have a mitotic zone, that is the nail bed. And in fact, this region is the actual germinative zone, which is highly proliferative and gives rise to the other cells. So, this skin which projects over the base of the nail, this is referred to as eponychium, and this portion which is in relation to the free edge of the nail, this is referred to as hyponychium, and this is nothing but the nail bed with the overlying nail plate. So, in fact, this nail plate is nothing but the stratified corneum overlying this nail bed, which is the stratum spinosum and the stratum basale layer. So, this is about the appendage nail, and we have various anomalies in relation to this nail, like we have spoon shaped nails in case of anemia, we have clubbed nails in case of some respiratory diseases. Then there is a breakage of nails which can occur, this is referred to as oncholysis. So, there are various clinical entities, just to name a few I have mentioned one or two. Okay. So, that is about the appendages. Few words about the development. So, we have various aspects of skin which are developing from different germ layers. So, from the ectoderm we have epidermis, the hair, the nails and the associated glands. From the mesoderm we have the dermal portion of the skin developing, and from the neural crest cells we have the melanocytes. So, the only germ layer which does not give rise to any part of the skin is the endoderm. Now, just to mention a few differences between thick and thin skin. So, when we talk about thick skin, this is the skin which is in reference to the palms and soles, and the rest of the skin which is overlying the limbs, trunk, etc., they are referred to as the thin skin. Thick skin is hairless, whereas thin skin is hairy. So, we will list out a few differences between these thick and thin skin. So, the general appearance, thick skin is a little shiny and glabrous, that is hairless, whereas thin skin is very thin to look at and it is hairy. The thickness, of course, as the name suggests, is thicker in thick skin and thinner in thin skin. So, the number of layers may vary from 5 to 10 in thin skin to hundreds in thick skin. And the different layers, all the layers are present in case of thick skin and at a thicker rate, whereas in thin skin most of the layers are reduced in size, and stratum lucidum may be absent in case of thin skin. Even if it is present, it is reduced to such a layer that is it is negligible. The appendages which are associated with thick and thin skin are also very different. We find the pilosebaceous unit being associated with the thin skin. Sweat glands are of course associated with both. And we have a few specialty receptors which are more common in thick skin, like Merkel's cells. So, these are more prominent in thick skin, and also Pacinian scorpels are unique to thick skin. We do not find them in thin skin. In fact, thin skin is having more free nerve endings, etc. And coming to the examples, like I already said, thick skin is present in the palms and soles, and the rest of the body is covered by thin skin. So, these are the few differences between thick and thin skin. So, this is the most interesting aspect of skin. So, we have innumerable clinical implications in relation to skin. Just to highlight a few, I am projecting a few interesting ones. So, this is a typical case of burns. So, you can analyze burns or classify burns as to various degrees in relation to the amount of skin which is being involved. So, if only the superficial layer is inflamed with no destruction, it is referred to as a first degree burn. The next, the epidermis may be lifted off, and it may slightly extend into the dermis. So, both these are various degrees of second degree burns, and if it reaches the dermal zone and much below that, that is the subcutaneous region as well, it is referred to as the third degree burn. And in extreme cases, you can call them as fourth degree burns, where there is a typical charring of skin which can extend up to the bone. So, this is a typical case of charring which was seen on post-mortem. So, when we classify or when we have to estimate the amount of burns, we generally follow the rule of 9. So, the face, the head and neck region has 9%, both the upper limbs have 18%, that is 9 each. The front of the trunk has 18%, back has 18, the limbs are again 18 and 18. So, all that constitutes about 100%. So, roughly it is in multiples of 9. So, we classify or assess the rate of burns based on this rule, that is the rule of 9. So, the next clinical entity we can talk about is excessive keratinization. So, we were talking about psoriasis, where there is excessive production of keratin associated with excessive epidermal turnover rate. Okay. So, there is excess, everything is in excess, the cells are produced in excess, even the keratin filaments are produced in excess. So, these conditions can result in conditions which can present as warts, corns or psoriasis, which is very common in these elbow regions or in the back of the neck, that is upper back region or scalp, etc. So, wherever they are more liable for friction, the chances of psoriasis in those regions are higher. So, all these conditions, there is excessive keratinization, that is overproduction or turnover of cells and overproduction of keratin. So, what determines the color of skin? There are various factors. It is not only the melanocytes, it is also the amount of blood flowing through, then the external factors and the amount of myoglobin present. All these will to some extent determine the color, but the mainstay is of course the melanocytes, and like I said, the melanocytes are mostly constant in number. It is only the amount of melanosomes which are present within the melanocyte that determine the color. So, in case there are zones of depigmentation. So, this is a typical case of vitiligo, where there are patches of depigmented skin. So, either the melanocytes are not present in this region or they are not functioning. There is another case, extreme case where there is total depigmentation of the body. The condition is called as albinism. So, here it is an autosomal recessive disorder where the enzyme tyrosinase, which is present in the melanocyte, is not active. So, there is complete depigmentation of the body. Now, this was deficiency of production. When there is excess of production of melanin, we have the other extreme. So, here we can have moles, nevus or melanomas. So, melanomas can either be benign or malignant, and they can be associated with hair as well. Even the nevus also, they are aggregation of these melanocytes in certain spots. Even freckles are also the same thing, aggregation of these melanocytes causing darker pigmentation compared to the rest of the body, and when they are associated with hair, that is the nevus, it is called as a hairy nevus. The skin can also be involved in certain infections and inflammations. So, this is a typical example where the skin is infected by the shingles or herpes zoster. And like I said, the distribution pattern of these infection is like 2 or 3 adjacent dermatomes. So, by just looking at the distribution of these lesions, you are able to arrive at a diagnosis that it is shingles. The other infections include acne, which are inflamed sebaceous glands, and more so you find in the forehead, around the mouth, etc. Okay, so, these are nothing but infected sebaceous glands. And we also have an infection by the lepra bacilli, which can affect the typical spinal nerve and cause sensory loss along the dermatomes. So, there are segmental sensory loss over the skin. So, these are some of the infections which can affect the skin. And the other clinical anatomy related structure entities to skin are the squamous cell carcinomas, the basal cell carcinomas, etc. And we can also talk about the skin grafts, and skin grafts are being collected in skin banks, and they are widely used especially for burns patients as of late. So, this was in brief about the skin. So, in today's session regarding skin, we studied that skin is a part of the integumentary system. It is the largest organ. There are 2 types of skin, that is the thick skin and thin skin. The layers of skin include the epidermis and dermis, and the cells which are present include the keratinocytes, the melanocytes, the Langerhans cells and the Merkel cells. So, with this we conclude skin and we move on to the next layer, that is the subcutaneous layer, which has the superficial fascia and the fascia beneath that, that is the deep fascia. So, what is fascia? So, fascia is nothing but a band or sheet of regular connective tissue, and it has many bundles of collagen fibers. So, it is present as a band or a sheet of connective tissues zone, which is made up of predominantly collagen fibers. And where is it present? It is present beneath the skin, then it is present around the organs, and it also is present separating the different muscle fibers. So, what are the types of skin? We have superficial fascia, we have deep fascia, and we have subserous fascia. So, subserous is very minimal in presence. So, it is predominantly the superficial and deep which we are concerned with. So, coming to superficial fascia, so this can be synonymously used with hypodermis or the subcutaneous region. The general features of superficial fascia include, so it is nothing but a layer of loose connective tissue with variable amount of adipose tissue. It is able to expand. It acts as a packing material between the structures. It acts as a storage area for fat and water. It also allows for free movement of the skin from the underlying structures. and it serves as a pathway for the neurovascular structures. Also, it plays a vital role in thermoregulation depending on the amount of fat available in the tissue. So, these are the general features which are common to superficial fascia in any part of the body. So, in fact, they are just acting as a packing material, helping in thermoregulation and forming a pathway for the neurovascular structures to pass from the deeper structures to the superficial structures. Now, what are the modifications of these superficial fascia? So, in certain areas it is ill defined. For example, in the dorsum of the hands and in face and neck, they are all very ill defined. It is well defined in case of scalp, palms and soles, and it is very distinct in case of the abdominal wall, perineum and limbs. In fact, you can see stratification in the lower abdominal wall, like you have a differentiation in the abdominal wall fascia, perineal fascia and in the thigh region. The distribution of fat varies from either being absent or in excess. So, depending on whether you need excess packing material, we have the distribution of fat varying in different zones. And the other important entity in case of superficial fascia is the presence of a subcutaneous muscle. Now, this panniculus carnosus is a continuous sheet of subcutaneous muscle which is present in lower animals, but in humans these muscles are left behind only in certain areas like the face and palmaris brevis. Elsewhere it is regressed. So, these are muscles which do not have any attachment to the bones as such. One end is attached to the skin. So, it is present in the subcutaneous zone. So, this is about the modifications of superficial fascia. Moving on to deep fascia. So, what are the features of deep fascia? So, this is a dense, fibrous, tough, inelastic connective tissue. So, superficial fascia was a very loose connective tissue, whereas this is a very dense, tough and fibrous connective tissue. It is devoid of fat and it is avascular. So, there are no blood vessels which are going to supply this deep fascia, and it is richly innervated by sensory receptors. And how does it respond to the sensory input? Either by contraction or relaxation, either by increasing, that is addition or reduction of size, or by changing the shape or remodeling, or by change of composition. So, if there is any stimulus acting on this deep fascia, these are the 4 different ways in which the deep fascia responds. It can either shrink, expand, change its shape, etc. So, coming to the modifications of deep fascia, it is absent in the anterior abdominal wall. It is ill defined in case of face and thorax. It is very well defined in case of limbs, and these are places where there is extra thickening of the already existing deep fascia. So, those include the intermuscular septa, which intervenes between the muscle fasciculi and gives origin to the extra muscles. Also, deep fascia forms the coverings of the nerves, muscles, arteries and glands. In case of nerves, they form different levels of coverings like the epineurium, perineurium and the endoneurium. Similar with the muscles, you have epimysium, perimysium and endomysium. It forms the tunics of the arteries, and also it forms the fibrous capsule of the glands. In addition, it forms the tendon sheaths and the fibrous flexor sheaths. Retinaculae are bands of connective tissue or deep fascia which plaster the tendons at a point, and they prevent it from bowstringing. So, we have external retinaculae, flexor retinaculae both in upper limb as well as in lower limb. We also have aponeurosis and raphe, which are modifications of deep fascia. The other modifications include ligaments. These are structures which are connecting two bones. We have interosseous membranes, that is between the bones like the radius and the ulna in the upper limb, the tibia and fibula in the lower limb. The other modifications include the joint capsules, the synovial membrane, as well as bursae, which are present in order to reduce friction. So, all these in total are nothing but modifications of deep fascia. So, like I already said, subserous fascia is not a very important fascia compared to the other two fascia. It is just present in relation to the serous membrane as a thin sheet, and it is more flexible compared to the deep fascia. And it contains variable amount of fat, and it is ill defined in the thorax, but well defined in the abdomen and pelvis. So, this is a very minimal fascia which is present closely in relation to serous membranes only. So, we do not talk much about this subserous fascia. So, coming to the clinical aspects or the clinical importance of these fascia. So, into the superficial fascia or the subcutaneous zone or the hypodermis, several injections can be given which require very slow absorption rates. Okay. So, wherever you require a very slow and delayed absorption of drugs or hormones, we can inject them into the subcutaneous region. So, these include injections like the insulin, which is regularly given for diabetes, the local anesthetics, and also the low molecular weight heparin. So, all these require delayed and prolonged action. So, they are given into this superficial fascia. What about the deep fascia? So, deep fascia is important because it forms tough fibrous sheath and it helps in compartmentalization. So, this compartmentalization, it will limit the spread of infection from one compartment to another, and by localizing this source of infection, the mass spread is limited, and also by determining where the infection is present, we can treat if suppose we are talking about clavipectoral fascia, if the infection is below, it will project into the neck. If it is present above, it will project into the limb. So, by determining where the infection is spread, we can localize the infection too, and the presence of deep fascia limits or demarcates the infected zone from a non-infected zone, and it limits the spread into other areas. And there is also a functional significance regarding deep fascia, because it is surrounding a functional group of muscles, like the flexor group of muscles are surrounded by one mass of a deep fascia, which act as a flexor group as a whole. So, similarly we have an extensor group. So, the kind of functional significance is provided by compartmentalization of different groups of muscles. So, this was about the deep fascia. So, in today's session regarding the fascia, we studied about what is fascia, the different types of fascia, that is superficial, deep and subserous. We saw their general features and their modified versions and the clinical implications of these fascia. So, as a whole, in today's session, we studied about skin and fascia as a part of the integumentary system. And like I said before, I do not know how much justice I have done, because skin is one big topic. All I have done is to outline the briefly regarding the layers of skin, functions, and the modifications of fascia. So, with this we come to the end of the session, and again I would like to thank the NPTEL SwayamPrabha team and the coordinator Dr. Vijay Sagar sir. So, intellectual growth, it starts at birth but ceases only at death. So, with that note, we will end the session. Thank you. Have a nice day.